Welcome to This Explainer. Today, we're diving into one of the most fascinating phenomena in all of biology, the body's ultimate balancing act. We're talking about homeostasis, the incredible machinery that quite literally keeps us alive.
It's a beautiful concept in the textbook, absolutely. But here is where we're going to make things really interesting today. We're not just going to sit here and talk about the theory. We're going to challenge how we actually prove this invisible balance exists in the lab. How do we measure it? How do we put a number on survival?
I mean, we hear it all the time, right? The body keeps itself in balance. But let's think like experimental scientists for a second. Bypassing the standard definitions, how do we actually measure dynamic equilibrium in a controlled setting? We can't just take the textbook's word for it. We have to design rigorous experiments, isolate our variables, and collect hard data to see exactly how an organism reacts to environmental stress. It's a duel between biological theory and practical scientific application.
Okay, well, before we hit the laboratory, we do have to establish our foundation. The textbook gives us this classic definition. Homeostasis is the regulation of the internal conditions of a cell or organism to maintain optimum conditions for function in response to internal and external changes. And guys, this isn't just a suggestion for the body. It is a life or death imperative. If we don't maintain these optimal conditions, our enzymes literally denature. They just fall apart. Metabolic reactions halt, and cellular survival becomes completely impossible.
Right, exactly. But this is exactly where theory meets praxis. The textbook hands you a neat little theoretical set point. For example, it says the ideal body temperature is exactly 37 degrees Celsius. Boom, done. But in a laboratory, humans are messy. A scientist must establish a literal control group just to find a working baseline. We have to continuously monitor normal physiological ranges for variables like temperature, pH, or blood pressure across multiple subjects before we can even definitively say whether a biological system has deviated from the norm.
And when those variables do shift out of range, this is how the body fights back. Tracing this feedback loop, we can methodically step through the process. First, a receptor detects a stimulus, so a change in a physiological factor. It sends that information through the nervous system to the central control, like the brain. The central control then instructs an effector, such as a muscle or a gland, to carry out a corrective action. It's brilliant. It perfectly illustrates how the nervous and endocrine systems communicate to continuously reverse any deviations and bring the body right back to its ideal value.
Let's apply this loop to a really classic example, thermoregulation. The central control unit here is the hypothalamus, situated safely right inside your brain. It acts as the body's master thermostat. The hypothalamus constantly integrates electrical signals from its own central thermoreceptors as well as peripheral receptors located all over your skin. Its primary job is to maintain a very narrow internal threshold of approximately 36.5 to 37 degrees Celsius. So, when the summer heat is on, your hypothalamus springs into action. Notice exactly what those effector actions look like out at the periphery in the skin structures. If the hypothalamus detects that you're getting too hot, it triggers vasodilation. Your blood vessels widen, allowing heat to radiate out from the skin. Simultaneously, sweat glands release fluid, and the physical evaporation of that sweat cools you down. Conversely, if you're too cold, we see vasoconstriction pulling blood away from the surface, and your skeletal muscles are triggered to shiver, basically generating and trapping essential heat.
Now, here is the really fun part for lab nerds. The real challenge is translating that internal physiological response into a rigorous scientific experiment. To test thermoregulation, notice how we isolate our variables in this experimental design. Our independent variable, the thing we as researchers deliberately manipulate, is the environmental temperature. The dependent variable we measure is the core body temperature of our subjects. And to make the data valid, we absolutely must have a control group kept at a stable 20 degrees Celsius ambient room temperature. This is how we separate the body's actual response from random environmental noise.
But how do we actually collect that dependent variable in a safe, precise way? In clinical laboratory settings, we use rigorous methodologies. We can measure temperature orally, tympanically through the ear, via the axillary method under the armpit, or rectally for high precision. But for modern, continuous data collection, say during an intense exercise stress test, we use data loggers. These electronic sensors allow us to process continuous physiological shifts accurately, without constantly physically poking, prodding, and stressing our human subjects.
Okay, taking the reins back for a moment, let's step away from temperature and look at an arguably even more complex system, osmoregulation. By definition, this is the control of the water potential and solute concentrations in your body fluids. It is a massive biological challenge. Water is the ultimate solvent of life. If an organism fails to balance water gain and loss, the results are catastrophic at the cellular level. Cells will either rapidly swell and burst from too much water or shrivel up and die from severe dehydration.
To understand how the body prevents that disaster, we have to look at the physics of the kidney. Osmosis dictates that water naturally flows from a hypoosmotic solution, where there are fewer solutes and more free water, across a selectively permeable membrane into a hyperosmotic solution, which is highly concentrated with solutes. Inside the kidney, blood undergoes ultrafiltration in the glomerulus. Then, as the filtrate travels through structures like the loop of Henle, salts are actively pumped out, and water just passively follows by osmosis. It's selectively reabsorbing exactly what the body needs. This physical movement of water builds into a complete, incredibly elegant hormonal circuit. Step 1. Specialized osmoreceptors, again in the hypothalamus, detect a drop in your blood's water potential. Step 2. Nerve impulses are sent down to the posterior pituitary gland, commanding it to secrete antidiuretic hormone, or ADH. Step 3. ADH travels right through the blood to the kidney, where it directly increases the permeability of the collecting duct epithelium. The kidney retains more water, returning it to the blood and leaving you with highly concentrated urine. Okay, that is an elegant internal mechanism, but I challenge you again, how do we verify what the kidney is actually doing? We don't just guess, we pull body fluids like urine or blood plasma and run precise laboratory fluid analysis techniques. We use qualitative reagents. The Benedict's test detects the presence of abnormal sugars, while the Burette test screens for proteins that shouldn't be slipping through the kidney's filter in the first place. We also use advanced colorimetry and chromatography, often linked straight to data analysis software, to measure the exact concentrations of electrolytes and fully quantify osmoregulatory function.
Now, it's absolutely crucial we draw a clear distinction here. Everything we've discussed so far, temperature, water balance, relies on negative feedback, which acts to reverse the stimulus and restore balance. But the body also utilizes positive feedback, which actually amplifies a change. Think of the release of oxytocin to increase uterine contractions during childbirth, or platelets rapidly clumping together to form a blood clot. While absolutely vital for survival, positive feedback is not homeostasis. It intentionally shoves the physiological factor further away from the set point to resolve a specific immediate event.
Which brings us to a rather dark, but scientifically necessary question. What happens to our laboratory data when these homeostatic mechanisms completely break down? Well, when systems fail, we are looking at severe pathology and disease susceptibility. If thermoregulation crashes in a freezing environment, hyperthermia sets in. The metabolism drops, dropping the temperature even further. If osmoregulatory hormones or receptors fail, we see metabolic disorders like diabetes. The set points are lost, and the data? It just reflects a system in absolute freefall.
So the crucial point is how this failure manifests in the numbers. Consider the secondary data derived from a study on diabetes insipidus. We are measuring the dependent variable of water permeability in the kidney, which relies on these physical channels called aquaporins. The healthy, wild-type subjects maintain a strong permeability of 196 micrometers per second. But look at mutants 1 and 2. Because of a genetic mutation in the aquaporin gene, the permeability drastically crashes down to 17 and 18. Without those physical channels, the kidneys simply cannot respond to ADH. Homeostasis is completely broken at the molecular level.
Let's contrast that pathological failure with the sheer immense power of a healthy mammalian system. Just looking at these osmolarity levels, normal human blood is kept incredibly stubbornly stable at roughly 300 milliosmoles per liter.
Exactly. And the magic is in the margins. To protect that 300 baseline, a healthy body can adapt dramatically. If you drink too much water, it can flush out highly dilute urine at just 70 milliosmoles per liter. But if you're stranded in the desert, the kidney can concentrate waste up to 1,200 milliosmoles per liter to aggressively save every drop of water. That is a massive dynamic range.
It really is. And that adaptability perfectly wraps up the theoretical side of our explainer. If we look at the foundational physiology texts, the core takeaway is remarkably clear. The body's many functions operate as to not deviate from a narrow range of internal balance, a state known as dynamic equilibrium. The ultimate imperative of all these complex cellular systems, the kidneys, the hypothalamus, the endless loops of negative feedback, it's all about sheer, unyielding survival through dynamic equilibrium.
And to conclude the application side, we must remember that our entire understanding of this biological survival is only possible through the power of the scientific method. We don't just observe, we test. Rigorous laboratory protocol, the precise measurement of distinct variables, and the objective processing of data. These are the actual tools we use to unveil exactly how life sustains itself against the chaos of the external environment.
Which leaves us looking to the future. As our environments push toward new, unprecedented extremes of heat and stress,
What are the absolute limits of human physiological adaptation? And more importantly, how will we design the next generation of experiments to find out?
keep thinking like scientists. Thank you for joining us for this explainer.